Okay, great. Welcome everyone to the first seminar of the Spring 2020 Biology, Ecology, and Evolution Seminar Series, also known as BEAS. This seminar series is organized by graduate students in the Applied Ecology and Biology departments. We are grateful for the departments and the college's support of this series as we continue to hold it in the virtual realm. Just to go over a few logistics that you've probably heard me talk about before, I would ask that you keep yourself muted during Dr. Beasley's presentation. And if you have questions while Dr. Beasley is speaking, please feel free to type it in the chat and we can save it for the end. And at the end of the talk, we should have 10 to 15 minutes for questions. You can either indicate that you have a question in the chat, raise your hand, or unmute yourself, depending on how many folks we have around, or Umbar will call on people to give their questions. I'll introduce Dr. DeAnna Beasley. Dr. Beasley is an assistant professor at the University of Tennessee at Chattanooga. She received her PhD in 2013 from the University of South Carolina. Her current research focuses on the effect of environmental change, such as urbanization, on insect systems. She involves undergraduates and citizen scientists in the research process. We are very excited and honored to have Dr. Beasley joining with us today. And with that, I will let her take it away.
Okay, well, thank you, Laura, for that introduction. And thank you, everyone, for coming to my talk. I'm so excited to talk to you. As some of you may know, NC State I consider it a second home. I know a lot of folks and made a lot of very valuable friendships here at NC State. So very excited to be back and talk with you about the work I've been doing. So my talk is geared towards undergraduate students, and so I'm very happy to hear that we have a number of students in the audience because often I'm asked to kind of talk about my origin story about how I got into science. And so I framed this talk around addressing that question, just to kind of give folks a little background. So I'll start with just showing this very nostalgic image of me when I was a grad student. This is me towards the end of my PhD at NC State. University of South Carolina. I went into grad school not with any preconceived idea that I'd be working with insects. My undergraduate background, I went to a small liberal arts college. I was a biology major thinking I was going to go to medical school. And then towards the end, I was like, I really don't want to go to medical school. I don't know what I'm going to do with myself. And I took an ecology course my senior year as an elective, and it just blew me away. Just the idea that you could quantify the environment and think about that in the context of how organisms are responding, how they're thriving, if they're surviving. It was a really compelling idea to me, and really exciting that there was actually a science that did that. So it shifted my thinking after my undergrad. And so when I started grad school, I was thinking, I want to study how the environment affects the health of organisms. And my advisor at the time was Dr. Tim Mousseau, and he had done his work with crickets primarily. He had since moved on to birds, but he had a hissing cockroach colony in the lab, and he just challenged me to develop a project using these hissing cockroaches. And I have to say, in the beginning, I was not keen on working with these organisms, but over time, over the course of my training, as I studied entomology, as I started asking more research questions, I really came to appreciate insects for what they can tell us about the environment. Actually, I'll go into more about the work I did at USC, but with insects, I really appreciate them for serving as a model for environmental change. We find insects across many different terrestrial ecosystems, including human modified environments. And and everything about them is sensitive to environmental change, their development, their physiology, for thinking in terms of immune function, since I came into grad school thinking about health, right? And even things like behavior were sensitive to environmental change. And so this actually worked out in terms of the types of questions I ended up studying. At USC in our lab, we were interested in studying the ecological consequences of the Chernobyl disaster. Now, for some students, you may not remember this. This disaster occurred before many of you were born, but for the rest of us, perhaps we might remember hearing in the news about the Chernobyl disaster. You're probably more familiar with the Fukushima disaster that occurred in 2011. It's the second most large-scale and severe nuclear disaster in our history. But just to kind of give you a brief background, so this disaster occurred in April 26th of 1986 when they were testing the reactor in terms of like how well it could cope with a partial shutdown. And in the end, long story short, a number of human errors led to reactor 4 exploding. And when it did, it just scattered radiation all over the local area. And if you can see here where my pen is, this is a pine forest near the reactor. The reactor's here. The radiation instantly killed the surrounding forest. And it turned the trees red. And so today it's referred to as the red forest. Here, this is a helicopter just flying lead into the burning reactor. They were just dropping lead into the reactor trying to suppress the fire. Eventually they sealed it with a sarcophagus just to kind of contain a lot of the radiation. But this was a significant disaster. About 49,000 people were evacuated from the city. And an exclusion zone was established where the radiation was just so hot, it was just dangerous for human life. But if you look here, here's the map of just kind of what happened to that radiation. The weather conditions and the wind patterns kind of scattered the radiation where you have variable pockets of radioactive activity. Some where you have fairly normal background levels, and others where, around the exclusion zone, it's pretty hot, right? And so initially, a lot of the cleanup efforts were done by cleaners, Chernobyl liquidators. Thousands of people who were responsible, civilian and military, who were responsible for actually going into these sites and cleaning up the radiation damage. So they were widely credited for containing and a lot of the immediate long-term damage from the radiation. But later, in the area, they started detecting higher levels of, for example, thyroid cancer in the population, as well as like childhood level cancers were being increasingly reported. And initially, there was some pushback in that the argument was, well, the reason why we're seeing this in human populations has more to do with social behaviors rather than the background radiation. For example, drinking too much, smoking, these are factors that are contributing to these incidences of cancer. And so my advisor, Dr. Mousseau, made the argument, well, if that's the case, then we would see these effects, in kind of wild populations here in the surrounding area. So my research was involved using insect populations, specifically focusing on the grasshopper. Served as a pretty good model because a lot of the grasshoppers' life cycle involved being in the soil, and that's where most of the radiation is today. And so my advisor collected eggs from the field across these various areas that had various background radiation levels, and brought them back to the lab. And so from there, I reared these grasshoppers in a common garden experiment, asking the question of what are the effects of these transgenerational effects of radiation on the grasshopper. And overall, what I saw was that, for grasshoppers and really whose parents came from really hot areas, very high radiation areas, tended to be smaller compared to grasshoppers from kind of low radiation sites. And that seemed also to be related to time to maturity. The faster you were able to develop and reach maturity, the larger you were. But if it took you a long time to reach maturity, then you tended to be smaller. And so this pattern between body size and reaching adulthood was a common pattern that we saw not only in insects, but in other organisms as well. We also saw evidence of mutations and deformities. So here are firebugs that were collected at some Chernobyl sites, where we're seeing here is a normal firebug, and then here are just the different types of deformities detected. So clearly evidence that the radiation was conferring some sort of stress on these organisms. A lot of the work in our lab was also done with barn swallows. Barn swallows have a very interesting life cycle where they migrate every year and they return to their natal site. So you can actually track individuals in terms of how, whether or not they return from migrating down to South Africa, back to East Europe where they were born. And so other things they noticed was, increased albinism, more discoloration, deformities in the wings and the like. They also showed that some of the fish in some of these rivers and lakes were also experiencing reduced body size. So, so this experience, this research kind of allowed me to think of the environment in terms of its impact on organismal health. Right. So since then, my research shifted. So after my graduate studies at USC, I came to NC State and kind of shifted towards looking at the effects of global urbanization, which is projected to add about 2.5 billion people to the world's population by 2050. And it was here that I really started to explore different insects. I started working with ants for the first time and looking at questions related to immune function and their behavior. I was really interested in their relationship with fungi and other disease-causing agents and how that varies. And I continued my work with periodical cicadas and started developing a citizen science project. And so I'll get into that in a moment. But here I'm starting to think more about the city as my place of study. And so here's my study site now. This is Chattanooga, Tennessee. It is, just to give you a little background, it is the only two-time winner of the Outside's Best Town Ever. The New York Times named it one of the top 25 places to visit. And this is in large part due to Chattanooga's, outdoor experience. If you are into rock climbing and any sort of aquatic activities, rowing is really popular. The pandemic kind of put a pause on many of these activities. We host the triathlon event every year. So this is a very popular place for outdoor sports. But Chattanooga didn't always have this reputation. So here's Chattanooga back in 1969. right? It was called the dirtiest city in America. Chattanooga has a very strong industrial history, where, you know, a lot of factories were in the area and they were just releasing tons of pollutants in the environment. There are stories of businessmen taking 2 shirts to work because the white shirt that they were wearing would turn gray after being outside because there was so much smog and pollutants in the air. So with the loss of some of these major industries, Chattanooga had to revamp itself, and it did, to its credit, to this, you know, more kind of outdoorsy, touristy place. However, we still see signs of this, you know, quote, dirty legacy, and it's a disproportional burden within the city. So here is the south side of Chattanooga, and it's considered a Superfund site, meaning that the area is so contaminated that it requires help from the federal government to help clean it up, right? This is a site that's located in one of the more poor, predominantly African-American neighborhoods. And that tends to be the trend, that your lower socioeconomic and your minority neighborhoods tend to take on the brunt of some of this environmental disparities. Right. So I became really interested in kind of understanding that variation in the city, and then the implications of that for the insect populations. What can they tell us? And just a quick background on urban environments, you know, in addition to the variation in pollution I mentioned, there's also these other factors that serve as unique ecological characteristics. The landscape tends to be fragmented. So we're talking about fragmented resources for some of these insects, a lot of paved surface, which has implications for temperature, high human population density, food waste, and then also higher amounts of invasive species, right? But a really, probably one of the more defining features of urban environments is that they tend to be hotter than the surrounding areas. And this process is called the urban heat island. And so that's due in part to the fact that, you know, we have more impervious surface, we have more cars, you know, releasing more heat, and we don't have a lot of vegetation in some parts of the city. But even here we have variation in temperature. If we zoom in at the city itself, we can see that there's variation in terms of how much heat is there. So this is Atlanta. And here at the bottom is the heat map. And so your darker colors represent hotter temperatures. And a study came out in 2020 where they were interested in understanding this urban heat island effect as a function of past redlining effects. So for those of you who don't know what that is, redlining is a systematic denial of various services by the federal government and local governments. And so you probably heard of redlining in the context of folks having a hard time getting a mortgage on their homes, like being able to build wealth through their homes. That's primarily how folks such as us, we build our wealth. And so through this systematic practice of denying mortgages and the like, a lot of these neighborhoods fell into disrepair. And so that means that a lot of these urban heat island effects is felt disproportionately by Black and brown citizens. And this study showed that Chattanooga in particular is one of the worst offenders in terms of the disparity between temperature in your formerly redlined neighborhoods and the rest of the city. In Chattanooga, it could be upwards of 10 degrees Fahrenheit hotter between the different neighborhoods. So for me,
Are they talking?
I'm sorry. Oh, sorry. So for me, this became a question of, you know, if we're expecting cities to expand and grow, and we're projected to, based on current trends, then looking at and exploring this disparity in the context of you know, the overall health of the city, the function, the ecological function of the city is important. Right. So here by 2050, we're expecting about, you know, 70% of the human population to live in cities. And if we look specifically at the Southeast part of the United States, we are expecting rapid urban growth in this area. So, so here, this was a study published in 2014 by Adam Tarando, where he showed that based on current projections, we could expect the city of Atlanta and the city of Charlotte, North Carolina, to merge into one large megacity. So, and with that, you know, with the growth of the city, you know, you have to think about the resources and the changes in the surrounding landscape to support that city. And so what are the implications of that for populations residing in the city? And so a lot of my thinking about research and my engagement with research comes with this framework of thinking about how do we build, or how do we create a more resilient, adaptive city? And we have a few examples of organisms that are actually doing very well in cities. So so well, in fact, that we kind of ignore them, right? Like we think of them as pests, you know, but they, you know, we overlook them to the point where sometimes we miss them until they do something really crazy, right? If we remember the raccoon that scaled the building all the way up to the roof of the building, you know. But I feel like they can tell us something about how they're adapting and how they're responding. And so, when I first came to UTC, I had a question just about looking at the overall diversity of different ants in the city. And one prediction would be that we might expect to see a decline in diversity, because as we move into the city, we start to lose our forest adapted ants, for example, and we start to see an increase in ants that are better adapted for city conditions. So we might predict that along an urban gradient that we might expect to see ant diversity decline. So I had an undergrad student, Hal Brooks, who did his honors thesis trying to address this question. We visited 3 different sites, a park, suburb, or street median that kind of represented the gradient along a city. And we just sampled for ants in these areas, and they counted the number of ants and you know, attempted to identify the different species of ants that they encountered. And overall, what they saw was, if we were just looking at the abundance of these species, that they did see a very small relationship in terms of the impervious surface, which we measured as urbanization, and the number of ants that they detected. Right, and most of the ants that we saw were these monomorium or tetramorium species that are found quite often in urban areas. I work with undergrad students a lot using ants as a model for kind of understanding these change. Even developed a course with where students were responsible for sampling for ants and developing research questions and hypotheses related to ants. And one of the projects that they did was inspired by Ant Picnic, which was developed in the Dunn Lab as part of Students Discover, where they laid out different food baits to assess food preference, dietary preference. And I did this every year in my ecology class lab for starting in 2016. So we had data that we could compare from year to year. And this was one of the results from 2017, where they found, unsurprisingly, that cookies were very popular with the ants. But they also showed in 2016 something interesting, where the ants also showed a preference for oil. Compared to the last year's group, oil wasn't at all nearly as possible. It was primarily cookie and sugar that was showing up in 2016. So this oil preference here was very interesting to me. And one of the things that I could tell comparing the 2016 class to the 2017 class was the 2017 class were selecting sites that had more impervious surface. While the 2016 students were heading out into gardens and kind of more wooded areas, the 2017 students were actually kind of staying close to the buildings, staying close to the classrooms. And so I had another honor student in the lab who was interested in understanding what may be driving this preference. And so we hypothesized that maybe one of the reasons why ants in these more urban areas, as indicated by impervious surface, they may be interested in having an oil preference because the food they would normally eat is not available to them. And so she thought, well, ants eat other insects, perhaps, or other arthropods, perhaps this has something to do about food availability. And so one idea was that as the prey availability declines in the urban habitats, you may see this preference for oil to increase. Alternatively, though, one can argue that you might see an increase in certain arthropods. We know, for example, from work done at NC State with Matt Pertone, for example, that arthropod diversity could actually increase in neighborhoods, and because houses can actually support a high level of diversity. So Itzel and I, we set up a study where we revisited the sites where we assessed diversity in the ants, and we laid out different qualities of oil. We created little feeding tubes that had, either like 1% oil or half a percentage of oil, or just like a full vial of oil. And we just we made a transect along these different sites and gave the ants a choice, in a sense. And we wanted to see does the quality of the oil or does the availability of arthropods determine the preference for fat, for oil. And so along these transects, we also set out pitfall traps. And Itzel, she was taking entomology at the time, and she was a very diligent student. She had, through these pitfall traps, she identified arthropods by families. And so I'm still actually trying to make sense of these results. I still am, I'm still puzzled by it because it's not what I was expecting. I'm just showing the 0.5% oil solution results, but it seemed like with the number of arthropods, that seemed like the preference increased. And so I'm still trying to make sense of what that means. If anyone has any ideas, I would definitely be open to hearing it. But it does kind of suggest perhaps that that ants may be supplementing their fats in another way, that it may not have anything to do with the availability of arthropods and maybe another supplement, for example. Okay, so I'm going to shift gears a little bit, talk about some work that I've done with cicadas. Periodical cicadas are another interesting insect in that they spend the majority of their life cycle underground and very much dependent upon trees and temperature. right? So, and the unique thing about these periodical cicadas is that they're only found in, this eastern part of the United States, right? The periodical cicadas, Magi cicada, comprises of about 15 broods, or different, and they emerge at different years. And they're very sensitive to urbanization, because urbanization fragments the landscape and reduces the connectivity among trees, right? In fact, we're scheduled this year to see brood 10. Most of brood 10 is primarily up in the northern part of the range here in Ohio, Indiana, and some parts of Maryland and Pennsylvania. We might see some of brood 10 in the eastern part of Tennessee. So, but I'm honestly a little skeptical. If we do, I would be a little surprised. But it is a fascinating phenomenon if you folks have not heard of the periodical cicada emergence. But one of the things that I was interested in is kind of going back to the effects of urbanization on body size in cicadas. So a study showed, for example, that cicadas show variation in body size along a geographic range. This is referred to as reverse Bergmann's rule, right? So Bergmann's rule suggests that, for mammals in particular, we tend to see big body size in colder environments, right? And then as the environments get warmer, you see a decrease in size, and so you can track this along a gradient. And so an example of this would be within the bear lineage, where you have your polar bear, which is one of the larger land predators on the planet, and then your smaller bear species in the rainforest area. Reverse Bergmann's rule, we tend to see this in your more ectothermic species, where you have smaller body size in your cold environments, and then bigger body size in warm environments. And so we see this a lot in insect species. And so I was very interested in seeing if we could see any relationship in how cities may be interacting with this pattern. We know that from that previous work that cicadas were also showing a similar geographic body range. And so we had an opportunity to study brood 2, which has one of the longest latitudinal ranges of the broods. So it ranges from North Carolina all the way up into New York and into Connecticut. And it was found in, it's in an area that's experiencing quite a bit of urbanization. And so this is a large range. And so I obviously could not do sampling on my own. And so this was a chance for me to work with citizen scientists. And so citizen science involves the collection and analysis of data related to the natural world by members of the general public. And I have done citizen science before, almost an opportunity to do so while I was in grad school. I worked with citizen scientists to track the brood 19 emergence when that emerged back in 2011. And so this is my first time being introduced to citizen science and being able to work with folks, not only in tracking these emergence patterns, but also educating and explaining to folks, the phenomena that they're experiencing, mainly reassuring folks that, periodical cicadas were not dangerous, and it will eventually stop. They will eventually stop screaming. And so with brood 2, I worked with Holly Miniger, who's now the Director of Public Engagement and Science at the Bell Museum. She coordinated with other citizen science. We developed Urban Buzz Citizen Science Project, where folks all over across the Brute 2 range sent us cicadas and sent them to the lab. And so with the cicadas, we measured body size, we measured the wings, looking at the width and variation in the wings themselves, asking questions about how does their size vary along their range, and if their location in the city tells us something about their effect on body size. And what we were able to show is that in the urban parts of, or the northern part of the range, in cities, the cicadas appeared to be larger compared to their rural counterparts, right? So we see evidence of the reverse Bergmann's rule in the rural populations, and we see that disrupted by cicadas located in cities. It appears, if we look at it by sex, seems like the males are primarily, we see this effect primarily in the males. What we can't say is what's causing this effect. It could be the urban heat island effect, with warmer conditions allowing the insects to develop longer and reach a larger size. It could also be other factors, such as the quality of the diet, right? So one thing that may be different is the quality of the urban trees compared to rural trees. So our study was able to show this pattern, but we can't, fully explain why this pattern, what the cause of this pattern. So still many more questions to explore with that study. Okay, so now I am, as you can see, I am not confined by any particular organism. I enjoy working with different types of insects. I've recently started becoming interested in exploring bees and looking at, again, looking at development and looking at morphology. And one of the effects of cities may have something to do with dispersal ability, right? Because cities are so fragmented in terms of landscape, we might expect shorter wings, which allows for better maneuverability around obstacles, but it limits dispersal ability. Longer wings, on the other hand, you have better dispersal ability. You can fly longer distance, but you may have limited maneuverability around obstacles. And so we did a short study looking at minor bees and looking at their wing structure and seeing if the shape of the wings and the size of the wings varied along an urban gradient. So we collected these wings, we collected these bees across different parts of the city, capturing again that impervious surface range. And in this case, we didn't see that that change in wing shape here. But I'm still interested. I'm still interested in exploring that question. Minor bees, they're solitary. They tend to stay fairly close to their resource, and so it may be a matter of, they may have the adaptive response just to stay put. So we're interested now in exploring that in honeybees. So I recently took on a grad student who's interested in exploring the health and the wing morphology of honeybees. And we are working with the Tennessee Valley Beekeeper Association, and we're tracking, the plan is to track honeybee health across Chattanooga and see if there's a relationship between where they are in the city and the health and quality of these wings. So this is a study. We just got RRB approval on it, so we're very excited to get going with it again. We had to shut down due to COVID, and so very excited to see this project pick back up again. So okay, well, that is all I have. I know I threw a lot at you folks. My research is very far-ranging, but the overall theme, I think, is the interest in urban ecology, and then just kind of exploring how the variation in the city, how that impacts these insect populations. And so with that, just want to thank all my undergraduate students and researchers for their hard work, and I will open it up to questions. Thank you. I'll stop the
Thank you so much, Dr. Beasley. It was a wonderful presentation, such well-rounded presentation, and your work with insects and the variety of it is amazing, really.
Thank you.
Yes, let's take it on to questions. If you have any particular questions, you can let us know through the chat, and then I'll call on you so you can make your question known. I see one from Sarah. Sarah, if you want to unmute and ask your question, or if you would prefer me to read it for you.
I can ask my question. I actually wrote like a few questions, but they're related. I just wanted to say I really appreciate how multifaceted your research is and like working with all these different insect groups. My question is, do you ever do any comparative work looking at different orders? Also, is it difficult to keep up with so many different species? And then my final question is, do you have a favorite insect?
Yes, I do comparative work, especially with COVID. I had to switch to doing a lot of literature reviews. So, I like to go through the literature and just try to get a comparison in terms of, like, for example, how are different organisms, how might we expect them to respond to urbanization, looking at shape, looking at morphology. So yes, quite a bit of comparative work there, especially now that I have more responsibilities as an assistant professor. Keeping track of these different insects in these different projects can be a bit of a challenge. I've had to learn how to scale back, or how to delegate. So I now have a grad student, a master's student, and she's wonderful, and she's really, you know, kind of keeping the project going and engaging with the beekeeper. She's a beekeeper herself, so it actually ended up being a good fit for her. And then my favorite insect, I feel like it changes all the time. But right now I gotta say it's probably still the ants. The carpenter ant was the first ant that I worked with, Camponotus. When I came to NC State, I consider it like the teddy bear of the ants, and that it's big and it does not gonna bite you. And so I still have a special place in my heart for the ants. So thank you.
Thank you.
I see James has a question as well.
Hi, sorry. I was curious about the oils that the ants eat. Like if they maybe outside of the city get them from seeds or some other source that maybe for some reason isn't as available in the cities. I don't know.
No, that's an excellent point, and I think you may actually be on the right track there, because so we know that ants, you know, rely a lot on plant oils and seeds for that fat, and so that might be, you know, another question, rather than looking at the arthropods, perhaps looking at vegetation and seed availability in the cities. That's point. So that's an excellent point. Thank you.
We have Michelle with a question up next.
Hi, great talk. Thanks for coming today. I also had a question about the ants. I was wondering what type of oil you were using in your baits.
We were using olive oil. We felt like that was, you know, as close as we can, you know, get to, you know, what they might get in the field. And it's similar to what was used in Ant Picnic and these larger scale citizen science projects.
And then did you see like certain species would come to oil more often? Do you know like what those species were?
So in the city, we were seeing a lot of monomorium, and not as much, we were seeing Tetramorium, but not nearly as much as we were seeing Monomorium, and then also fire ants as well.
Thank you.
Dr. Green has a question.
Dr. Beasley, great talk. It was very interesting. Thank you very much for the presentation, and the breadth of material was very interesting. So yeah, I also had an end question, and you sort of alluded to this with the Bergmann's rule information and the city temperature. Is it possible that ants' nutritional demands change with temperature?
That was, yes, I would think so. So, you know, we know that, or, you know, there are some that might suggest that, you know, shifting towards a more fat-based diet in hotter, drier conditions may provide a more reliable energy source of some sort. But that is a very interesting approach in terms of just thinking in terms of variation in nutrient demands. Also, you know, we know that dietary shifts occur just based on the life stage of the colony itself, and so it'd be interesting to kind of explore that in the context of the city.
Yeah, looking at maybe some of the temperature data between your 2 years, between 2016, 2017, even with the urban changes as well, might give you some hint at maybe potential demands are different.
Right.
Yeah, very
Right.
cool.
Oh, I hear you. If it were hotter, perhaps that was why they're shifting.
On the one day versus another, yeah. And then that is also sort of influenced by the urban state as well, as you showed in the heat maps.
Right.
Awesome. Very cool. Thank you very much.
Thank you. Yes, that's a good point.
I see Elsa has a question too.
Hi, Elsa. You're muted.
Muted. Hi, it's good to see you and great to hear all the things you've been working on.
Thank you.
You've piqued all our curiosity with the ant nutrition thing, clearly. And my question, maybe I just wasn't paying attention, but I think when you described the experiment, you mentioned that you put out 3 different dilutions of oil, and then I think you showed the graph. It was the 0.5% showing that the abundance of ants at that percent increased with arthropod availability. But I was curious what happened to the other 2 concentrations. Like, were those ignored and the 0.5% was what everybody went to, but just in different numbers? Or was there actually a shift in preference among the 3 kinds?
There was a shift. So I didn't share that graph because I'm still like trying to wrap my head around what happened. It was weird. So like the 0.1 dilution, the preference went down. It seemed like for the 1% and the 0.1%, the preference went down, but it was a weak effect. So I don't know, you know, what we can gather from that. But it's because it completely was not what my hypothesis was. I'm completely confused and stumped by it. So I've been sharing this talk and sharing this data and hope that someone would be like, hey, Dr. Beasley, I have an idea, because I need closure on this study here.
Cool. Yeah, I certainly don't have the solution either, but yeah, I'm curious about if there's another way to look at preference between the 3.
Right.
And then the role of which species are involved in explaining any of it. But
right. So there could be, and we'll have to go back and look, anecdotally speaking, just from what I remember from the study, Monomorium seemed to be the dominant species that were coming to our baits. And so, but they weren't the only one. And so it would be interesting, I'm sure there's some dietary preferences even among species that, could be driving what we're seeing.
very cool. Thanks.
Thank you.
And I see Dr. Rice has a question as well.
Hey, Dr. Beasley, thanks very much. That was really, really interesting work and well presented.
Thank you.
It occurred to me, or I wondered, it seems like the dietary preference thing would be amenable to some experimental work where maybe you could rear ants on like a seed-dominated diet, arthropod-dominated diet, whatever you think the key variables might be in the dietary differences that are out there, and then expose them to the preference test and see if that can give you a key to how to interpret their behavior.
That's a good idea. We'll certainly have to explore that after COVID's done. And
you just need a bunch of citizen scientists with ant farms on their windowsills and send them different things to feed their ants.
Okay. I like that. That's actually a really cool idea. And then just kind of get this, collective data building, or I could just incorporate it into a class.
Your class would be better,
Right.
yeah.
No, that's a great idea.
Well, I don't see any more questions in the chat. So thank you everybody for all of your great questions, and thank you, Dr. Beasley, for giving us a little bit of your time today and for starting the seminar on such a strong note.
Oh, well, thank you all so much. It's like I
You're
said,
welcome.
NC State's like a second home to me. So it was such a treat being able to see everyone again. Miss you all.
So yes, I think we can bring the seminar up to a close. Stay tuned to your inboxes for the next seminar that will be coming up soon as well. Thank you, everybody.
Thanks, DeAnna.
Thank you. Bye, all.
See you.